So here we are in our second to last class period ever for biology. And for some of you guys sitting in the room today, or some of you sitting in the room tomorrow, this very well might be the last time that you ever sit in this classroom. All right. It's been an interesting ride, to say the least, through our COVID experience, but the end is near. The major thing between y'all and what could very well be probably one of y'all's longest Christmas breaks, right? I mean, some of y'all are going to have almost, 3 weeks? That's pretty sweet, right? And so you can definitely eke out a little bit more effort leading up to that, knowing that you have 3 weeks or almost 3 weeks of vacation, right? To rest and relax and to recover from all that was the fall semester. Now, as always, in our customary tradition, I would like to start class by looking at our checklist for the second to last time. Yesterday, it's sad and it's exciting, you know? I don't know about you guys. I'm ready for Christmas break. I think everybody needs a vacation break. All right, so yesterday, hopefully you've completed the practice EOC, because this was a wonderful opportunity for some of you guys to potentially replace some low exam grades. However, the deal was, for you to have that opportunity, it had to be done by last night, okay? And so for those of you that didn't turn it in, unfortunately, that ship has sailed for you, okay? Now, that doesn't mean that when we go over it, you shouldn't keep track of the correct answers, because those questions are going to be a great kind of guide in your preparation for the EOC. Now, for those of you guys that did complete it in time, when I go over it later, you guys will need to either write it down on a piece of paper or open up another document, and you'll need to write down the answer key. I'll be giving you 30 of the 61 questions, the answers. And then I will send back your submitted answers, and then you'll be responsible for marking the ones that you got right, correct, and the ones you got wrong, wrong. And then you will be responsible for submitting it, resubmitting it once it's been graded with your final score. And if that final score is better than one of your lower exams, I replace it just like that. But I'm also doing this to test your honesty, because I'm sure some of you guys have figured out with Google Docs, if you share it with somebody, have you guys looked at the editing history? You can actually pull up the history of a document and see exactly what changes were made when and on what date. So let me preface something. Let's say on your practice COC you made an 85, and you think, dang it, I wish I would have made a 95. You know, because there was that 170 on that exam that I made that, you know, I need to improve it. Having a 95 would be really nice. So maybe I'll just change out these answers so it looks like I made a 95. If you do that, what I'm going to do is I'll take your lowest exam grade and I'll replace it with a 0 for breach of academic integrity. So an 85 replacing a 75 is way better than me replacing a 75 with a 0, right? And so I'm trying to kind of teach you guys, like, what is the word? Morality and ethics, right? Just because that wallet's laying on the ground stuffed full of cash doesn't entitle you to rob that person blind, right? Just as if you lost your wallet, you would hope that someone would find it, contact it, and you'd get it back with all of the contents, right? Always think, if you were the other person, what would your expectation be? Okay. Now, what are we going to do today? I'm going to go over the EOC schedule with you guys, right? So you know where to be on what day and in what room and what to bring and what you'll need. And then we'll do a quick Kahoot on cells. And once again, by just participating in the Kahoot on cells, what are you going to get? 100. And so for some of you guys, this is going to be your saving grace, the Hail Mary into the end zone. But again, you have to participate. Yesterday, I had a student join it. They answered like the first 2 questions, and I guess they went and played Xbox, right? And then they sent me an email like, what the heck, man? Like, why aren't you going to give me credit for this? I said, because if I ran a marathon, if I said, yeah, I ran a marathon, come to find out I walked the first mile and then walked off the course and laid down to take a nap. Is that really running a marathon? No, right? So that person did not participate. Answering 2 questions and falling asleep or picking your nose for the next 20 minutes does not count. Then I'm going to quickly review mitosis and meiosis, and then I'll go over half of the practice COC. And then any remaining time that you have in class, you can be working on your study guide. Remember, your EOC study guide is due tomorrow night. Now, if between now and tomorrow you run into some pages that are a little difficult or challenging, please send me an email or a remind. I've had students send me pictures like, yo, Colin, what in the world? And then I literally send them an email back with the answers. I like, hey, look at this question. This is why, or check this out. Send me a link, whatever. Okay. Again, you need to have that done because that's another really, really easy grade. Okay. That'll kind of, I want to set you up going into this EOC so that you're confident and that you have like a cushion. Because remember, at the end of the day, I have no control over the EOC. I have no control over the percentage. I have no control over the questions, right? And it has been a heck of a semester, right? Full of challenges. I was really impressed with y'all's benchmark scores, right? Right before Thanksgiving. Y'all's average was not much different than prior semesters, right? And so I think you guys should be confident. Despite all the challenges, you guys have done really well, right? That's why we're doing the practice COC. And as we go over it today, some of y'all will be like, whoa, sweet, man. I did pretty well on that, right? And I haven't really hunkered down and studied yet, right? So hopefully that'll give you some confidence. These other assignments, like the study guide, that gives you some confidence, but also gives you a cushion. The practice COC, replacing a low test grade, confidence and cushion. All these little review games, the little Kahoots, confidence and cushion, right? Because remember what I said in the beginning of the semester, if you've had a 91 or a 92, if you've been sitting with an A average all semester long, and you take that EOC and you make an 82, which by the way, 82 on a cumulative, that's awesome. But an 82 that counts for 20% of your grade, what's that probably going to do to a 91? It's probably going to drop it. But you've been busting your tail. You completed the study guide. You did the practice COC on time. You don't have any missing assignments. You've been grinding all semester. There's a little button that I can click in PowerSchool. It's like manual override, right? Now, if you've been a little stinker and there's a long rap list of missing assignments and all these wonderful, easy opportunities I've given you to like make up for past mistakes you didn't take advantage of, and you've been riding a 70 all semester, maybe a 72, and you take the EOC and you make a 20, and that drops you down to like the 62, I'm just going to look at that button and think, woulda, coulda, shoulda, right? It's a hard lesson learned, but when presented with opportunities, multiple opportunities, multiple doors open for paths to success, and you just choose to ignore all of them, I take it kind of as an insult. And if someone were to insult you day in and day out over the course of, I don't know, 18 weeks, how likely are you going to be to stick your neck out for that individual? I consider myself an overly compassionate individual. That is my biggest personality weakness. I love all of you, and I probably give you way too much respect, right? Way too many second, third, fourth, and fifth chances, right? But there's a line that I need to draw in the sand, and that's that line, okay? All right, so let's go ahead and get started. Let's go ahead. If you go to your Google Classroom, the very first assignment at the very, very top is you'll see EOC exam schedule. Have you guys seen that? You probably saw it popped up. When you click on it, you'll see second page is for second period. Second page is second period. Hold on, y'all. I think I need to see who that was. In case it was an emergency. All right, so we are second period, so y'all will be taking your EOC on Tuesday, okay? So Tuesday of next week, and it is here at Patton in person. It doesn't matter if you're A day, B day, or virtual academy, or remote learner. Everyone will be here on Tuesday to take their second period biology EOC. Now, I don't want you to think, oh no, are we all gonna be all 36 of y'all crammed in this tiny little classroom? No way. So what we've done is we've kind of tried to split y'all up to make sure that we're safe and to make sure that we can maintain social distancing. So if you're an A-day student, you guys will be in here, okay? So this is where y'all will be, right? B-Day students, plus Mrs. Baker or Ms. Baker, you'll be in 702, which was Mr. Francis's old room. So it's still in this hallway. And then all my virtual academy students, you guys will be in 706, which is just right next door to my room. So if for whatever reason on Tuesday you're walking to school or coming to school and you're like, oh man, I don't remember what class, no stress. Because whose job is it to put you in the right place? Right? I'll be there. I'll be in the hallway. Hey, friends, long time no see. Right? And I'll say, hey, you need to be in that classroom. Hey, you need to be in this classroom. Now, if you want to make this as stress-free as possible, here are some hints. Arrive early. I can be an anxious person, and nothing revs up my anxiety like rushing and being late somewhere. I hate it. If I'm with people and they make me late, the whole time my heart's just thumping in my chest. I absolutely despise it. 5 minutes early to me is still late. I like to get places early enough that I can kind of scope out the scene, maybe get a drink of water and find the bathroom. Right? That's just how I am. Stress kills, ladies and gentlemen. And it really does kill performance, right? You've probably experienced this. We were so stressed out and so overwhelmed, you could barely even think straight. That's not how you want to be. The building opens at 7:30 on Tuesday, so I would suggest trying to be there early, okay? We're going to get rocking and rolling at 8 o'clock. So you rolling in at 7:55, it's going to be a little stressful because when you show up, we got people showing up late. When you guys show up, you'll need to have your laptop, your Chromebook. You have to have your Chromebook. So make sure that your Chromebook is charged and make sure you have logged out of your Chromebook. When you guys come into these different rooms, there'll be seating charts. Like, all right, you sit here, you sit here. You'll sit down, you'll get your laptop out, and then I'll come around, or the other teachers who are adminning your exam will come around, they'll collect your laptop, they'll sanitize it, they'll log in, pull the exam, hand it back to you after it's been sanitized. And then you just sit and wait. I don't know about you guys, but if I were taking an exam, I would like to be early, and I would like to be the person that at 7:45, I'm sitting there at my desk looking at the starting test screen, be like, all right, do I need any water? Do I need to go to the bathroom? I would like to be twiddling my thumbs 10 minutes into exam rather than walking in at 7:59 and in a whirlwind panic trying to get your computer set up while everyone else is relaxing watching you. That makes me anxious thinking about it. Just try to be there early, okay? Make sure your laptop's charged and make sure you've logged out. It'll just make the whole process a whole lot easier. And you have to make sure that you've logged out and you're charged, and you want to make sure that you have your Chromebook. You can't use your own laptop. If you want to have some reading material for afterwards, as long as you're not reading a biology textbook. So if you want to bring a novel or something, or a magazine or a comic book, that's perfectly fine, okay? And so make sure to check to see where you're at. Again, if you don't remember, we'll direct you in the right place. Everyone needs to take the exam. Now, if you are currently quarantined and you're thinking, oh no, I'm going to miss my exam because I'm quarantined, I can't come to school. Don't worry. If you're currently quarantined and your quarantine time period kind of bleeds over into next week's exam schedule, I will be sending you dates and times for your makeup, because we have quite a few students that will all be taking the exam at a later date due to their quarantine length. All right, so that's the exam. That's next week. And for us, second period, that's Tuesday. All right, let's take a look at our cells Kahoot. Okay, real quick, a review of the cells. Everything from transport to organelles and types of cells. If you guys would take a second to join this, and I think I do need to return that call. So while I'm waiting for y'all to join, and remember, participation is key to the success in developing your EOC cushion. So I'll give you guys a second. I'm gonna pause. All right, back in action. So, all right, we're still waiting on a lot of y'all. Still waiting on 4 of y'all. Let's go. Let's do it. If you want that credit.
Wait, what'd you cook?
Breakfast burritos. Yeah, I roasted some potatoes, some radishes out of the garden. I put some Mizuna on it. So Mizuna is like an Asian green that's kind of like a nice peppery taste. and then some homemade salsa that we made from our tomatoes this summer and peppers. And so I used chicken eggs and some of our quail eggs.
Well, now I'm hungry.
And then some shaved sharp cheddar. It was quite delicious. Made for an excellent Thursday because I like to celebrate Friday Eve,
Yeah.
right? It's the second greatest day of the work week, you know? All right. Come on, slow pokes. Let's do it. Come on.
I like Saturday Eve, and I like Saturday.
Saturdays are great, but I honestly think Fridays are my favorite because I do like work, but I love the thought of the weekend too. So there's something magical about a Friday, you know? Saturdays are pretty good. I love Saturday morning, just waking up and knowing that you have the entire day to do whatever you want for most weekends.
Well, Saturdays are usually reserved for clean couple times.
Oof. See, I save that for Sunday.
I have church, and then I have cheer practice after that.
Gotcha. All right, ladies and gentlemen, let's get ready to rock and roll. What's that red dot?
Oh, we now see it.
All right. When a cell uses energy to transport ions against the concentration gradient. So what kind of transport requires energy in the form of adenosine triphosphate, ATP? That would be active transport. Excellent work. And remember, active goes against the grain. So we're moving things from low to high, kind of against their natural tendencies. Which of the following substances would cross the cell membrane using facilitated diffusion? Remember, facilitated diffusion moves big molecules. So which of these molecules is probably the biggest and would actually need like a protein channel to simply diffuse through the membrane? So facilitated diffusion is still passive. No energy is needed. The molecules are still going from high concentration to low concentration. They just might be really big and they might need an opening or a protein channel to move through. So which of these looks the biggest? Easiest way to do it is count the atoms. Yes, that'll be glucose. In fact, the proteins that allow the channel for glucose are actually called GLUT proteins, or glucose transport. Predict what would happen to a cell that was placed in hypertonic solution. What would happen? And remember, a good example of a hypertonic solution is soda, full of salt and sugar. So if I took any cell and I placed it into a beaker of soda, what would probably happen? Good. Now, here's what I want you guys to do on Tuesday. You'll get a scrap sheet of paper, and you can write whatever you want on it before the exam starts, right? You can give yourself little notes. Again, one of those weird things I thought about in high school. I drew it on my paper. We had EOCs even back when I was in high school in biology. And I drew a can of soda, right? Don't laugh at my drawing, right? And I named the soda hypertonic. I just drew that in the corner of that little blank sheet of paper, and then I knew for the rest of the exam, anytime I thought about hypertonic solution, I thought about soda, and soda has lots of sugar in it and lots of salt. I think, I'm not for sure, but I'm pretty sure 2 or 3 sodas exceeds your daily recommended sugar and salt intake. So if you didn't eat anything all day and you only drank 2 cans of soda, you've already exceeded the USDA's recommendation for salt and sugar. Now, soda is going to make you real fat, and it's probably going to give you hypertension. And the reason for that is because salt and sugar suck. They taste delicious, but they wreak havoc on our bodies because the excess salt and sugar in soda actually dehydrates us. It sucks water out of our cells. And what a brilliant marketing scheme. Coca-Cola and their ice cold glass and their commercial, everyone's hot, and they drink their cola and they feel nice and refreshed. So then you're hot and you're thirsty and you're like, man, a soda sounds great right now. And they won. Because they know if you're thirsty and you grab a soda, if you drink that can of Coca-Cola, by the time you're done, you're even more what? Thirsty. And then you'll drink a second and then a third. And then instead of the small soda, you get the extra big gulp. And then they say, hey, let's market it to fast food. Because when you're eating fast food, you're eating potatoes, which are sugar, covered in salt, dipping it in ketchup, which is a sugary salt sauce. And then you're eating your chicken nuggets, which are covered in salt. And they're covered in batter, which is sugar. And then you're dipping it in a barbecue, which is sugar and salt. And then you're so thirsty from eating your salty French fries and your salty nuggets with the salty ketchup and the salty barbecue that you can't even imagine drinking that 12-ounce soda. And you think, it's only $1 more to get the mega gulp. So I'm going to go ahead and get that 48-ounce Coca-Cola. And then by the time you slurp that down, because boy, you're thirsty, on the drive home, you said, man, I'm thirsty. You stop by Food Lion, you pick up a 12-pack of Coca-Cola. And the cycle just continues and continues and continues. And there they are sitting on their mountain of gold. There's a reason why those soda cans and all those companies usually use things like reds and blues, because they psychologically kind of drawing you in. I love talking about soda. What a wonderful marketing scheme. Probably kills more people per year than like heroin, to be honest. The leading cause of death in America are all related to obesity. Americans are literally eating themselves in the grave. The movement of water. What do we call that? Think about water oozing in and out of a cell. Yes, it's osmosis. I just think osmosis ooze. I think about water oozing in and out. Yeah, you can play along in the chat if your internet's kind of funky. No worries. Yes, I will send you something later. All right. In what direction will water move if I place a cell into a hypotonic solution? Hypo. And a good example of a hypotonic solution will be pure distilled water. So what would happen if I took a cell and placed it into a beaker of distilled, pure, 100% water? Yes, the water is going to go into the cell. And that's why if you were lost at sea for weeks, maybe even have drank your own urine a couple times, but don't do it more than twice, because by the third time, the urea concentration is toxic. You can actually do it once or twice if need be. Probably wouldn't be very good. But you'd be so dehydrated, a cruise ship comes by, they pick you up, they bring you in, and you see a pitcher of ice cold water, and you think, I want that. You grab the pitcher and you start chugging it, and you may or may not have just killed yourself after surviving for 3 weeks on the open ocean, because all of that fresh water would do what? Go rushing into your shriveled, decrepit, little dehydrated cells, and they would expand too rapidly and potentially pop, right? So that would be not good. So more than likely, what would be the first drink they'd offer you? Yeah, they'd probably give you like a Coke or Gatorade. You'd be thinking, what the heck, man? Right? I want water. And they have to slowly introduce the water back into your system. That's why if you're really dehydrated and you go to the hospital, they put you on an IV. And the IV actually contains what? Yeah, the saline solution, which is kind of weird. You're like, what the heck, man? You know? In what direction will water move if the cell is placed in an isotonic solution? Yes, the net movement is zero because iso means equal. And so growing up when I was young, my dad was a big, college football player, really into lifting weights. So I kind of grew up in the gym, and I was kind of a meathead myself, which I know is kind of hard to believe for most of my younger life. And, I was like, Arnold Schwarzenegger was kind of like my hero. And so I used to read all kinds of stuff about lifting weights and exercising. And I learned about exercises and contractions, right? So when you contract your muscle, right, and you bring a muscle shortens, that's concentric. Eccentric is when the muscle stretches. And when the muscle is held and doesn't shorten or lengthen, it's held at an equal state. That's an isometric contraction. So I learned that way back when, and I always think isometric means equal. People always say, I don't have time to do 1,000,000 crunches to have abs. Well, I tell them isometric contractions. I'm doing abs every day, all day, even while I'm teaching. I'm doing it right now. Now, if I were to do like a traditional crunch, the concentric would be me crunching in, and the eccentric would be me stretching back out. So how in the world do you work your abs while you're just sitting there in class? You just tense up. An isometric contraction means that the shortening and lengthening are equal. So it's just like you kind of sitting there, sucking in your belly button and just holding it tight. And see how long you can do that while you're moving around and talking and typing on the computer. I don't know if you guys ever realize the way that I talk is like kind of fast. And then eventually I'll take breaks and I'll breathe for a second. So what do you think I'm doing the whole time that I'm talking? I'm in an isometric contraction. So by the end of the day, I'll have been working my abs out for 3 periods of 90 minutes. Now, how many of y'all have ever sat down and said, all right, I'm going to work my abs out for the next 270 minutes? I don't do that, but I do it during school. It's isometric, so it means equal. There's always a way to cheat the system. But again, it relies on science. Which cell process is shown in the image below? So you can see these particles kind of being engulfed or absorbed by the cell. Now, the cell is probably going to have to invest some energy in the form of ATP to get the cell membrane to change shape like this so that it can engulf and bring all these particles into the cell all at once. That is endocytosis. So think of endo like endoskeleton, right? So it's like inside of your body. Which is an example of active transport. Something that sounds active. It sounds like we're going to need energy. We're going to need ATP. Look for like a verb or a noun that can be also used as a verb. But be careful, make sure to read the entire answer because there are two answer choices that both have nouns that can be used as verbs or adjectives that can also be used as verbs or imply movement. Yes, this is the best answer right here, the sodium potassium pump. The movement of water, that's osmosis. Now, osmosis is the diffusion of water, that's passive. Carrier proteins move ions with a concentration gradient. That's like facilitated diffusion. The molecules are still going from high to low. They just need a protein to help. So despite it saying carrier and you're thinking, hey, that sounds active, that's still passive. The best answer is the sodium potassium pump. It was outlined in our PowerPoint. I went over it in my recorded lecture way back when. And this is probably the most famous of all active transport pumps. In fact, this is how your brain generates an electrical signal to trigger neurons to release neurotransmitters, which eventually trigger acetylcholine release under your muscles, which triggers contractions. And there's like a neat little electrical impulse throughout your muscles. And that's why tasers work, right? So the taser like artificially produces that electrical impulse. And what happens to all your muscles, right? They get all tense. So that's the sodium potassium pump. That's also why you need sodium potassium in your diet. What do we call sodium and potassium and calcium if you were to take it in like a nutrient supplement? It's in Gatorade. Electrolytes. Yeah, electrolytes. I wonder what they deal with. Generating electricity inside of your body, right? It's a part of this active transport. Now, why do we need electrolytes? Because unfortunately, what happens to some of the sodium, the potassium, and the calcium when we exercise? Yeah, we sweat it out or we pee it out. So we need to replace it. And you know when your sodium potassium pump's running out of sodium potassium, right? Because you get cramps, right? The muscle contracts and it won't release. So if you guys get Charlie horses a lot, you're like laying in bed at night and you're like, oh my God. And you feel like your hamstrings like twisted up or in the bottom of your foot, you're dehydrated and you need electrolytes. Cells begin bursting when placed in a solution. So more than likely the solution or the environment is what? If the cells are exploding. Think hypo-hippo. I wonder how many of you guys are tucking in your belly button right now and maintaining isometric contractions. And it works for all muscles. Which process is used by cells to excrete wastes? So when your cells need to get rid of waste products, or when certain cells like neurons need to release neurotransmitters, or maybe even the alpha and beta cells of the pancreas need to release insulin or glucagon, they're going to use this active process to release or to allow molecules to exit the cell actively. That would be Exocytosis, because that is an active form of transport that allows lots of large particles, molecules, and things to be released. You thought active transport, yeah. So active transport is not wrong, but it's not specific enough. It's not the best answer, if you will. Facilitated diffusion. Which of these best describes facilitated diffusion? Remember, it's facilitated, which means assisted, but it's still diffusion. So it's still a form of passive transport, and passive transport tends to go from high to low, kind of going with the flow, if you will. Yes, it moves with the concentration gradient. It just needs some help. Which accurately describes the sodium-potassium pump? It's a very famous form of what kind of transport? This form of transport requires a source of energy that's typically produced by some form of respiration, whether it's anaerobic or aerobic. This is produced. The amount might vary, anaerobic producing less of this, aerobic producing more. It's still important for the sodium-potassium pump. Yes, moving these things in and out using ATP. Which of the following is not an example of passive transport? So which of these would be considered active? And your cells would need ATP to accomplish this. 3 of these are passive and require no energy whatsoever. Yes, endocytosis. Because facilitated diffusion, diffusion, and osmosis are all passive, so things are going from high to low, no energy needed. Endocytosis, cells changing its shape to bring something inside. If a cell is 80% water and the outside is 90% water, what's going to happen? So we got our cell, and inside the cell, it's 80% water, 80% solvent, and the environment is 90%. What in the world's going to happen? Where does the water want to go via osmosis? And, I know that's 80% water, the remaining 20% would be the solute, right? So that could be sugar or salt or really anything dissolved in the water. And outside would be 10% solute. Yes, the water is going to want to go from high to low. The water will rush in, and our little cell will swell and expand. So that tells us that the outside is hypotonic, okay? Because there's more water on the outside than there is on the inside. Hypo means below, so the salt concentration or solute concentration of the environment is below that of the cell. Animal cells and plant cells have what in common? So what would this little kitty cat have in common with a corn plant? And although cats are domesticated, they were domesticated for a much shorter period of time than dogs. That's why they're more fiercely independent than dogs. Yes, animals and plants are going to have a nucleus because they're both eukaryotic. All cells have cytoplasm and ribosomes, and they both have a mitochondria because they're both eukaryotic and they both need aerobic respiration. Prokaryote or eukaryote? Look at the picture. Is this a prokaryotic cell or a eukaryotic cell? Remember, pro is no, eukaryote is the do-karyote. So prokaryotes don't carry a lot inside. Eukaryotes do carry a lot on the inside. Yes, this is a prokaryote. How do I know? Well, I don't really see any major organelles. I don't see a nucleus. I see this nucleoid, but then I see circular DNA, and I say, circular DNA is prokaryotic. Eukaryotes have linear DNA, like the cool little chromosomes that we see and draw all the time. And then another dead giveaway for me is the name of this flagellum. It says bacterial flagellum, and I think, the only type of organism that is generally classified as prokaryote or is classified as prokaryote is either going to be bacteria or archaeobacteria. So if we're talking about prokaryotes, it's always bacteria. Everything else, like, fungi, protists, plants, animals, all those funky things are all eukaryotic. A prokaryote keep my mask up. Pin it under my glasses. My face is moving too much. Yes, has no nucleus. Prokaryote is the no nucleus. No nucleus, no organelles, no large size, no complexity. Which of these is not a eukaryote? So which of these organisms is not a eukaryote and would be classified as a prokaryote? Yes, excellent. Fungi would be like mushrooms and mold and yeast. Protists would be like algae and euglena and amoeba. And then plants, well, plants. Whereas the bacteria would be the only group belonging to the prokaryotic cells. Prokaryote, eukaryote. Look at the picture. And then once you decide whether it's a prokaryote or eukaryote, could you maybe identify what kind of eukaryotic cell that is? Could you maybe identify some of the organelles? Excellent. Look at that, 100%. Yes, this is a eukaryote. I see a nucleus. I see all kinds of membrane-bound organelles. And I know this is a plant because it's green. That helps, right? We got a cell wall. We got a cell membrane. I see this massive vacuole to store water because plants need water. It's one of the necessary reactants for photosynthesis. The vacuole also uses the water to keep the plant rigid. And then I see our little chloroplast right here for photosynthesis. What does a eukaryote have that a prokaryote does not? So which of these will be found in eukaryotic cells and not prokaryotes? And you might be thinking, my goodness, Mr. Collins, enough with the prokaryote, eukaryote. Well, this should be a pretty good indication that being able to differentiate these is going to be important. Guys, these are the simplest questions that you should not miss when it comes time to the OC. Yes, all eukaryotes will have a nucleus and prokaryotes will not. Pro is no. What does a vacuole do, right? Why are they so big inside of a plant cell, right? So you'd have your big cell wall and it's kind of square. Then you have your cell membrane inside and this massive vacuole. It takes up a majority of the plant cell. Like what in the heck? Why? Why is it so important? Especially for plants. Yes, water storage. Because plants just can't get up and go find more water. I mean, their roots can grow deeper into the ground, but they need to have the ability to store water for photosynthesis. Ribosomes. What are they? What do they do? Where can you find them? I can see the mRNA here being read by the ribosome, and the tRNA is delivering the appropriate amino acid. The amino acids are getting linked together with peptide bonds, forming a large protein, right? So ribosomes synthesize proteins. And this whole process of the rRNA and the ribosome, reading the mRNA, reading it 3 nucleotides at a time, there's a codon, slamming on the right amino acid. This whole process that occurs in the ribosome to synthesize proteins is known as translation. Right? We're translating the mRNA into amino acids. And the order of these amino acids is determined by the order of these codons, and those codons are determined by the DNA from which the mRNA was transcribed. The phospholipid bilayer, what in the world is that? Where are we going to find it? Made of phospholipids. All cells have this. Helps control what enters and exits. Really involved in passive and active transport. Is selectively permeable. Doesn't let everything in, it doesn't let everything out. That is the membrane. All cells have a nucleus. True or false? Dang, those answers just ripping in. False. A vast majority of life on Earth is prokaryotic, believe it or not. There are more bacteria in a spoonful of soil than there are animals on planet Earth. So, despite our familiarity with the larger macroscopic organisms that are all eukaryotic, not all cells have a nucleus. Bacteria do not. In fact, if I were to put you on a scale and weigh you, right? I weigh about 195 pounds, maybe. What if I told you that a vast majority of my mass is actually attributed to bacteria? You have more bacterial cells on the outside of your body and on the inside of your body than you have human cells in your body. So that should make you feel kind of special, right? You're like a walking, talking ecosystem. All these bacteria need you. What is the purpose of the mighty mitochondria, which will be found in all eukaryotes, by the way? The mighty mitochondria uses aerobic respiration to catabolize glucose in the presence of oxygen to produce this, which would be essential for active transport, the ATP. Excellently, write down our winners. Excellent work, y'all. And then I want to quickly just review. Let's see here. Cell division before we go over the practice COC. And I'm going to keep this real short, real simple. Again, I'm just trying to like wake up some of those neurons. All right. Later. Tomorrow. There we go. All right. So, cell division. All cells have a cell cycle, which usually results in them dividing. And we can break this down into two main categories because we've got our prokaryotes, right? Our single-celled bacteria that lack a nucleus. And then we've got our eukaryotes, which would include, algae and fungi and plants and animals, right? Well, when prokaryotes divide, they use what kind of division? It results in identical copies of bacteria. Binary fission. There you go. Binary fission. Fission is division. And so bacteria have the ability to clone themselves with this really neat process called binary fission. And this is what allows bacteria to kind of develop a genetic resistance to things so fast. Because if you kill 99% of the bacteria, all it takes is one single bacteria to survive. And he might survive because he's a genetic mutant, immune to the antibiotic because of his genetic mutation, and then it clones itself, right? And so really rapidly, you end up with a population of bacteria that are identical, and they all have that same gene for resistance. Eukaryotes, not so much. We need to look at eukaryotic organisms, especially multicellular. So we're going to look at me. We'll use my body as an example. And we can look at all these eukaryotic cells. We can split them into two main categories. The somatic cells, right? The cells of the body. And then our germ cells, the cells of our reproductive organs, right? In the case of a male mammal, it'd be testes. Female mammal will be ovaries, right? And we would agree that they both have different life cycles, right? There's definitely a different end goal for those two types of cells. Now, I don't care what type of cell we're dealing with. The cell life cycle always starts with interphase. Now, what happens during interphase? Interphase is the longest phase of the cell cycle. And during interphase, the cell is going to go through a period of growth known as gene 1. And then we're going to make copies of our DNA. All right, we got to make sure that we have extra copies of DNA because we're going to divide, and we want to make sure that these new cells have what they need. And then we're going to make some more proteins, and we might grow a little bit before we divide, right? And so that after interphase, the cell will move into the next phase of the cell cycle. But that's where it really matters, whether it's somatic, a body cell, or a germ cell, like the cells of a testis or an ovary. The cells of my body, such as the cells in my toes, are going to experience what type of division? Mitosis. So in mitose, you've got skin, bone, muscle, and nerve. They're all somatic cells, and they'll undergo mitosis. Now, what about in the mammalian testes or the mammalian ovary? What happens in those special sperm and egg making cells? What kind of division do they experience? Not mitosis, meiosis. My egg is ovulating, so insert sperm. And that reminds us that, oh yeah, it's dealing with sperm and eggs, it's dealing with sexual reproduction. And meiosis is a whole heck of a lot different than mitosis. So let's take a look at mitosis. What does mitosis start with? Well, we'll start with a somatic cell. And if we're talking about human somatic cells, all human somatic cells are diploid, right? And so what in the heck does that mean? Well, diploid cells, I can also write it as 2N. What this means is the diploid cell has 2 of each type of chromosome. And that's because I got half from my mom and half from my dad. So I have 2 copies of chromosome number 1. One I got from my mom, one I got from my dad. And so for a human being in this diploid somatic cell, how many chromosomes would you find in any of the cells located in my toes? Whether it's skin, bone, muscle, nerve. How many chromosomes? Can you remember for humans? We have 23 types, and I got 1 from mom and 1 from dad. So that would make for how many total? 46. Good. Yeah, so my somatic body cells would have 46. Now, after interphase, it starts mitosis, which would be prophase, metaphase, anaphase, and telophase, right? And so during mitosis, the chromosomes kind of line up and get split and pulled to different sides. And then eventually mitosis is over. But if we were to stop right here at telophase, the cell would still be kind of stuck together, look like a peanut. And so we still need cytokinesis to happen. Cytokinesis is the separation of both the cell membrane as well as the cytoplasm. And as a result of cytokinesis, after mitosis, we have 2 daughter cells. Now, the daughter cells produced by mitosis are interesting because they are identical. Not only are they identical to one another, they're also identical to the parent cell, right? So if I cut my toe, the skin cells will undergo mitosis to replace those damaged skin cells with identical skin cells. And since they're identical, they are also diploid somatic cells. So how many chromosomes would be in these 2 new cells? The same as the original. Okay. Now, what about meiosis, right? And so in the mammalian testes, you got these specialized cells, spermatocytes, and the female ovaries, you have oocytes. And they start, these spermatocytes and the ovary cells, right? The cells of the testes and the cells of the ovary, they're still a part of your body, right? We only call them germ cells because they have a different life cycle, but they're still diploid, right? So these initial cells still have a full set of human chromosomes, right? And it's not until meiosis begins that the number starts to change. Now, remember, meiosis happens in 2 phases, right? The first division is prophase 1, metaphase 1, anaphase 1, telophase 1, right? That's meiosis 1. And then it divides a second time, prophase 2, metaphase 2, anaphase 2, telophase 2, right? Now, some magical stuff happens during prophase 1 and metaphase 1, but we'll get there in a second. Right? So we divide once in meiosis 1, and then it divides a second time in meiosis 2. But again, if we stop there, everything would be kind of stuck together, and we'd have 2 peanut-looking things. And so we still need cytokinesis, the separation of the cytoplasm and the cell membrane. And what we end up with are not 2, but 4 cells. Now, since we divided not once but twice, the daughter cells or the products of meiosis are quite strange. And you will see questions exactly like this. They'll be like, what are the products of meiosis? Well, what are they? I see 1, 2, 3, 4 cells instead of 2. And these 4 cells are not identical. Each one of these is unique. And I'll explain why here in just a second. And they're not just unique, they're haploid. each of these would only contain 23 chromosomes, half of the original. So they are haploid. And I could write that as just simply a lowercase n. So they only have one of each type of chromosome. And these are no longer somatic. They're no longer even germ cells. They're known as gametic cells or gametes. And gametes would be things like sperm or egg. Now, each one of these sperm contains a unique set of genetic information. So each one of these sperm has the potential to create a completely different human being, okay? And that is the important and beneficial aspect of sexual reproduction, is because two parent organisms can produce lots of offspring, and there's no telling what you're gonna get, right? It's like you're shuffling the deck each time and pairing them up and producing all of these wonderful variations that we see. And variation is important for natural selection and evolution, for the success and survivorship of various species. Now, how do we get these wild and wonderful different sperm? Well, during prophase, you've got crossing over, right, where chromosomes kind of swap alleles, they swap genes, and that mixes things up. And then during metaphase 1, when they line up in the middle, they don't line up necessarily with their designated partner or on the right side. They kind of independently assort. They all line up randomly, and it's kind of like a shuffling of a deck. So both crossing over and independent assortment increase genetic variation. And then we have no idea which one of these sperm is going to be the sperm to fertilize the egg. And so we have random fertilization again, which also increases genetic variation. Now, sometimes during meiosis, the number of chromosomes doesn't split evenly, and sometimes the sperm ends up with 24, or sometimes the sperm ends up with number 22, and that's called non-disjunction, and that's not good because non-disjunction can cause things like Down syndrome, right? If you inherit one extra chromosome at the 21st pair, you end up with trisomy 21, which is what causes Down syndrome, all because meiosis didn't happen properly. All right, that's it for my little mitosis meiosis. I'll save that. And now what I want to do is no worries. I want to take a look at y'all's practice EOC. So what I would like for you to do right now is either get out a sheet of paper, or you can start another Google Doc, because I'm going to be going over these answers, and I want you to write down an answer sheet. Yes. Yes. Make it quick, because you're going to be missing some very important stuff. And then you're going to use this answer key later to go back and check and grade your practice COC. So you'll have like two Google Docs, one with your answer key, and then one with the practice COC you already took. And yeah, oh yeah. And the reason I want you to have the answer key is so that you can take the practice COC again. You maybe wait till Sunday and see, okay, how much do I remember? Okay. And so again, it's right here, practice COC. I will return your submitted answers for your practice COC for you to grade as soon as we're done with this. And I'm going to go over half of it today and half of it tomorrow. And I know you may be thinking, oh, Mr. Collins, cut it out, let us go. Guys, I'd rather you do your studying now so you have time to like kind of let it soak in than for you to try to wait and cram, or you'll say, yeah, I'll study this weekend. No, you won't. You're a procrastinator. A lot of you are. That's why some of you guys are still turning assignments that were due 3 weeks ago. What happened to that, right? So we're going to do it now. All right. Question number 1. This is an animal cell. Yeah, no duh. I know it's not a plant because it's got like a circular shape. And it says, where are the cell instructions located? You submit your study guide tomorrow. It's not due till tomorrow night. And if you did it paper and pencil, you can always bring it the day of the EOC. All right. So where are the instructions? Where's the DNA? That would be in D, the nucleus. So number 1 is D. Y is pointing to mitochondria. W is pointing to ribosome. And then X, the cell membrane. Number 2, which can be found in eukaryotic cells but absent in prokaryotes? That would be your membrane-bound organelles. So number 2 would be D. Number 3, which would be the least likely benefit of a transgenic crop? Remember, transgenic is just like GMO. So which of these things would you not want as a genetic engineer? I don't know about you guys, but I don't want to decrease the resistance of my corn crops to viruses. I don't want them to be more likely to be infected, right? So D is a terrible thing to do to a crop. The other 3 is great. Make it last longer on the shelf, increase its nutritional value, and decrease its susceptibility to pests, which means that it would be now less susceptible or less likely to be eaten by beetles and bugs. So 3 is D. 4, which is the basis for a phylogenetic classification. Think, what in the heck is that? So phylogenetics, we're classifying animals based on, DNA evidence and what they look like. We're trying to make an evolutionary tree, right? We're trying to figure out the family history of species, right? And so it would look something like this, right? So maybe you've got like a line and another line up here and then a line over there and a line down here and then maybe a line that comes way off here, right? And you got these different species, right? And so what are we trying to figure out here? We're trying to figure out the evolutionary relationships, right? So the answer would be D. Now, will you see a question asking what is phylogenetics? Probably not. Will you see a question like this where you're given a phylogenetic tree and I ask, which of these 2 species is most related? The length of the branch matters. The shorter the branch, the younger the separation. So which of these 2 species is most closely related? They probably have the most DNA in common. A and B. Because did they separate a long time ago? No, man. Look how short their branches are, right? They've only separated maybe a couple 100,000 years. Who would A's most distant relative be? Would have the least in common with A? Yeah. Because how long ago did those 2 species separate? Man, forever ago. And over the course of that forever, they've been accumulating differences, right? So you read it just like a family tree. Over time, an antibiotic will no longer be effective on some bacteria. Why? Guys, this has nothing to do with us. Because this could be happening in a petri dish, right? So A and B, throw it out. So what do you guys think, C or D? If an antibiotic kills 99% of bacteria in a petri dish, 1% will survive. And what will that 1% do? Yep, they're going to pass on that genetic resistance to the next generation. So that is C. The surviving bacteria are resistant to the antibiotic and they'll continue to breed. You guys ever heard of MRSA? MRSA? Really nasty skin infection. It's called MRSA because it's methicillin resistant Staphylococcus aureus. Wrestlers get it, football players get it, people in nursing homes get it, daycare workers get it. And once it gets inside of you, it doesn't really respond to antibiotics. If you ever get a little cut and the cut gets kind of swollen and tender, and so you put some antibiotic cream on it and it goes away. Well, MRSA, you put the cream on there, nothing. It gets bigger and pussier, and then maybe you even get a fever. You go to the doctor, they give you antibiotics, it's still not working, right? A lot of times they have to cut it out, right? So my brother's got a big scar on his forehead. He had a cut on his head, or maybe even a pimple, we don't even know. And he got a MRSA infection from his football helmet. And they had to like cut the tissue out of his forehead. So he's got this really crazy scar on his head. All right. 6, to enter or leave the cell. What do we got to get through? Yeah, the plasma membrane. It's another name for cell membrane. So 6 is D. 7, how do I know I'm looking at mitosis? Well, I only see like one division. Are these cells in order? Heck no, man. This is all jacked up. This is prophase. This would be technically prometaphase, but we don't really worry about that. We got metaphase, anaphase, telophase. Which of these is metaphase? That's B. Word number 3. How do I know? Meta, think middle. Meta is middle. Ana away. Telo, it looks like it's tearing. 8. Which environmental factor is most likely to cause melanoma or skin cancer in human beings? UV radiation. B. Ultraviolet radiation can cause mutations in our skin cells, resulting in melanoma, which is actually the most common form of skin cancer, or common form of cancer. Now, can X-rays cause mutations and eventually result in cancer? Yeah, but there's not that much X-ray out there, right? So it's not likely to cause.
People don't like get an X-ray.
Yeah, right. And if you do get an X-ray, they try to protect you as much as possible, right? Why is it when you go and get an X-ray of like your abdomen or your like leg, they make you wear those like lead undies, the lead diaper, the lead crotch shield? They're trying to protect your gonads. They're trying to protect the ovaries and testes because if they cause a mutation in your testes and in your ovary, well, you know right away. But could it affect what? Yeah, you might have a baby later and be like, whoa, the baby's got squid arms, right? Because of the mutation in your sperm. Not really. I'm just being ridiculous. Greenhouse gas emissions are increased as a result of which of these activities? Well, hybrid cars are great because they reduce the amount of gasoline burned, so that's not right. Alternative energy like wind and solar, that's gonna reduce greenhouse gases. Regulating smokestacks is gonna help, but the more people who drive cars, the more greenhouse gases we're gonna see. So the answer for 9 will be D. 10, when do cells use anaerobic respiration, right? So anaerobic, like intense exercise or holding your breath or yeast in a barrel that's sealed. That's going to be when oxygen is absent. Does anaerobic respiration produce a lot of energy? Heck no, man. That's like a backup plan. That's not good. When you go anaerobic, you know that your workout is almost over. 11, which best describes a change in the nucleotide sequence of DNA after exposure to radiation? So if we mess up DNA, if we switch it up, we change it up, that's a mutation. What in the heck is a codon? That's that 3 nucleotide sequence in mRNA. 12, which scenario best describes an occurrence of geographic isolation? Remember, geographic isolation, animals get isolated on or as a result of geographical features, separated by oceans, rivers, mountains, canyons, and they live in different areas, so over time they become different. Which of these matches that? Yes, 10 is B, sorry. 10 is B, 11 is C, and 12 would be C. Finches, which are birds living on different islands, different food sources, will slowly become different. 13, which explains why all cells in an organism have the same DNA, but different structures and function. Doesn't matter what kind of cell you get from a crime scene. As long as you get a cell, if it's muscle, skin, bone, it's going to have the same DNA in it, right? So all the cells in my body have the exact same DNA. So then why is my muscle cell different than my brain cell? Why is my brain cell different than my skin cell? Is because there are different genes in our bodies that can be turned on and off. And so my skin cells are skin cells because the skin cell genes are turned on and those cells are making skin proteins, whereas muscle cells, muscles, because muscle genes are turned on. So the best answer right there is C. And that whole process that happens in the embryo of those different cells becoming different, that's cell specialization or differentiation with genes turning on and off. Which will most likely happen to the rate of photosynthesis if light intensity increases? So if I slowly start giving a plant more and more and more sunlight, what happens? Initially, photosynthesis increases, but doesn't increase forever. Eventually, it levels off, right? Because their rate of photosynthesis is limited by the number of leaves and the number of chloroplasts. It would reach a maximum. So 14 is C. Or no, sorry, 14 is D. 14 is D. Sorry, I got tired, dead little eyes. They kind of look the same. Yes. 15. Which role do enzymes play in cells? They are biological catalysts that decrease the amount of energy your body needs to invest to get that reaction started, right? And so they are C. They decrease the activation energy. All reactions have a little bit of investment that needs to happen, and enzymes just make our cells need less energy to get started. 16, which process creates a DNA fingerprint for forensics? So you collect some DNA from a crime scene, you chop it up with restriction enzymes, you put it into a gel, you zap it with electricity, and you get those cool bands that separate smaller fragments, travel less distance, big fragments travel far. What do we call that process? Gel electrophoresis, right? All right. So are you going to see something exactly like this? Probably not, right? They're getting a little simpler. right? But these dichotomous keys, you will see one. You will see one of these kind of questions. Look at it. No wings, right? I don't see any wings. So I go to question 5, so I can skip all this. Terminal appendages resemble pincers. Terminal appendages are straight. Terminal means at the end. Like if you have a terminal illness, that's going to be your end, right? Or the terminal at an airport is where the plane like stops. That's the end of your journey. You get off and on at the terminals. Now, appendages, those are like ligaments, right? Things that stick to and off the body, and pincers, like crab claws. So what do you guys think? Do we have straight appendages or are they like crab-like claws? They're straight. So we go to 6. Are there 2 terminal appendages or 3 terminal appendages? I count 1, 2, 1, 2, right? So I only see 2, right? So what do we think? Diplora, all right? So that would be B for 17. 18. Which is a sustainable way of managing renewable resources? So first and foremost, it has to be renewable, which means it comes back very quickly. And we need to use it sustainably. In other words, if we're going to use it, we have to replace it at the same rate. Fossil fuels, nope, throw it out. Those are non-renewable. Water for crop irrigation, that is crap. Irrigating crops is not very sustainable, right? Because that means that you're growing plants in a place where they probably shouldn't be. Plant a new tree when you harvest a tree, that's about as sustainable as it gets. Cut one down, plant one. So right there in that moment, has the number of trees changed on planet Earth? Nope. That's very sustainable. So 18 would be C. Here we have a undifferentiated, unidentified stem cell. And we're looking at all the different specialization paths or cell differentiation paths it could become. So, all these cells over here at the end all have the same DNA. They have just different genes that have turned on and you've transcribed and translated different proteins. So how is this possible? Well, that's because each of these cells at the end have different genes that have become activated, right? So 18 should be A. That is the best answer. Oh, wait. Sorry. 19 should be C. Wait, no. Yeah, 19 is A. Sorry. Get ahead of myself. 20. Okay, which 2 species are most closely related? Again, who's close and whose branches are shortest? X and Y. So 20 is A. Who is Y's most distant relative? You go down back in history and a long, long time ago, right? This would be like the great-great-great-great-grandpa, right? How can the use of pesticides on crops impact the environment? So, not yesterday, Tuesday. If you spray a bunch of pesticides, a lot of it's going to end up in the water, end up in the soil, and so it can eventually do what? Get magnified, and it can accumulate in the food chain. So the best answer here is D. Right? It's not going to reduce the diversity of crops because you're killing pests. Is it going to create more pesticides to feed on crops? What does that even mean? That answer choice is just wild. It increases the reproduction of non-resistant pests? Heck no, man. Pesticides are going to kill all the non-resistant pests, but it will accumulate in the food chain. Some of you guys probably right now have substantial concentrations of pesticides in your blood, right, from the foods that you're eating. And antibiotics from the chicken that you maybe ate from Chick-fil-A. And some of you who eat a lot of tuna. I mean, I eat a good bit of tuna sometimes, or used to eat a lot more. So I probably have mercury in my body. 22. Local farmers have been using a pesticide for years. It used to work. Which is a negative impact that could have on the ecosystem? Now, when you spray pesticides, they usually kill a lot of related pests. So if you use rat poison to kill rats in your house, what other animals might you need to be afraid of? Or what other animals might you need to be cautious of being exposed to rat poison? Rats are mammals. So any of your cats and dogs, they eat it, they're dead. They eat the rat, they're dead. So when you spray a pesticide for like insects, and if there's a beetle eating your crop, it might kill like your bumblebees or butterflies. It might kill pollinators. So 22 is B. 23. Some cells maintain a higher concentration of potassium. Hey guys, potassium, we're maintaining, we're working hard. 23 is active transport.
Danielle Crowder, Michael Dale,
24.
Glorimar,
How does DNA help to form proteins? Well, DNA has the instructions
Chris
or the sequence, right? So DNA, the sequence, becomes mRNA, and then the mRNA gets translated into protein. So the best answer for 24 is D. 25. Which is most likely to affect an antelope? Remember, antelope like deer population as a density-dependent factor. So deer or antelope, you learned this in earth science. We talked about in AP. These are factors that are intensified by density. So the more densely populated they are, the worse this becomes. Which of those problems is going to become worse for a densely populated group of deer? Disease, right? More dense, like New York City, 25,000 people per square mile. Forget that place. I don't know why anyone want to ever live there. And it's no kidding why the coronavirus is out of control there. Because you walk a mile to the street, you're in the same area as 25,000 people. That's more people in Morganton in one city block, right? You can't breathe in the city of New York without inhaling someone else's fart, sneeze, or cough. It would just be impossible, right?
Wait.
Okay, 24 is D, 25 is A. 26, which stage of interphase is, are we gonna make copies of DNA? That's C, synthesis. 27, which are components found in many lipids, but not nucleic acids? What are lipids made out of? Glycerol and fatty acids. 27 is C. Amino acids make up proteins, monosaccharides make up carbohydrates, and D is just crap. 27 is C. 28, during the dry season, the Galapagos Islands, the number of certain beaks starts to change with the available food. And then what are we looking at? As an environment changes, the animals change, the plants start to change. What do we call that? Some survive and reproduce, some die and don't. That's natural selection. Now, what if we were doing the picking? What if you're like, that bird's pretty, let's feed it. I don't like that bird, it's ugly, let's kill it. That's artificial selection. That's how we got dogs and cats and pet birds and things like that. Okay, 29. Which process produces mRNA or messenger RNA? That would be the transcription of DNA. Once you've transcribed DNA into mRNA, then we can translate the mRNA with tRNA and RNA, rRNA, into the amino acids that will become a protein. 29 is D. And last but not least, not D. Which process would use energy to move stuff? So we got to use energy. We got to use ATP. So 30 is C. All right, ladies and gentlemen, as I had mentioned before, now you have the first 30. Yes. 23 was C. Now that you have the first 30, when I return, right? When I return your little practice COC, you can grade the first 30 questions. Hey, could you maybe go back and look at the last 30 again tonight? Maybe you felt rushed and you didn't have time. Yeah, because when are we going to grade them? Tomorrow, right? And then tomorrow you'll grade the whole thing. You'll calculate your score. And if it's good, right, you'll say, Mr. Collins, I'd like to replace a low test grade. Now remember, don't try to cheat the system, right? Because if you try to cheat the system, I can tell, because I can tell when you make your changes on your Google Doc, right? So I can see that if today you go back and change half of the answers from the first 30, then I will award you a 0 to replace your lowest test grade, which I know most of you probably have most test grades higher than that, right? So this is like just where being an honest human being really helps out. Okay. If you missed the turn-in date, unless if you have a really good reason why, then I might be entertain the idea. If not, you're simply you kind of missed out on that opportunity to replace. Okay. All right. Now tomorrow we'll review DNA with another Kahoot for another 100. We'll go over genetics, and then we'll go over the last 30 of this. And again, you have to be here so that you get those answers, so that you grade it, so you can get credit for it, right?
So we just don't turn it, or do we turn it in and it's just the?
All right, hold on. I gotta answer my phone. See you guys.